Welcome to the deep dive. This is where we get into the latest, most impactful papers in longevity research.
Yep, and our mission really is to be your guides.
Exactly. We cut through all the noise to find those essential nuggets of knowledge for you, the biotech researcher, the curious mind.
We're always looking for the stuff that genuinely pushes the field forward.
Especially those findings that might just need you to, change your model, rethink things a bit.
It's funny, science has this rhythm, doesn't it? Some weeks, wow, you get these truly groundbreaking, paradigm-shifting discoveries.
Right.
Other weeks, it's more about solid, incremental advances. Still critical, the building blocks.
The foundation.
Precisely. And I'd say this week, well, it leans more towards the latter. But still, there are some really valuable insights here for anyone in the field.
Absolutely. So let's dive into this week's batch. We know true novelty, that huge leap, is rare, but even these smaller steps build towards the big breakthroughs we're all working towards.
Couldn't agree more.
Okay, let's kick things off. This first paper, it involves a really intriguing reclassification.
Ah, yes, this one caught my eye too.
It's from July 17th, under Albayram, Natalia Oleinik, and Besim Ogretman from the Medical University of South Carolina, published in Aging Cell.
Good journal.
Title: Beyond the Pseudogene. P17 permit as a mitochondrial trafficking protein linking aging and neurodegeneration.
So a pseudogene that's not.
Exactly. They found that this thing we dismissed as a pseudogene, RPL29P31, actually encodes a functional protein. They named it permit.
Okay. And what does permit do?
It's a mitochondrial chaperone. It mediates ceramide transport between the ER and mitochondria, which is crucial for mitophagy.
Mitophagy.
Yeah.
The cell's quality control for mitochondria, super important in aging.
Right. And here's the kicker. Losing permit actually speeds up sarcopenia, that's age-related muscle loss and neurodegeneration in their aging models.
Wow. Okay, what's fascinating here really is this idea of reevaluating parts of the genome we kind of wrote off.
The so-called junk DNA.
Exactly. If you connect this to the bigger picture, it suggests there might be this whole reservoir of hidden regulators of aging just sitting there.
A whole new place to look for therapeutic targets.
Potentially, yes. It really challenges that traditional view of the genome structure and function.
It's definitely compelling. But, as always in science, validation.
Oh, absolutely. While the finding itself is fascinating, we need more. Its broader impact hinges on further validation.
So what does that look like?
Well, specifically, we need confirmation in mammalian longevity models. We're talking proper in vivo lifespan studies.
Like overexpression studies.
Yes, permite overexpression and also tissue-specific knockouts, especially in aged mammals. That's how you really see the organism-wide effect.
Got it. Okay, so from genomic surprises, let's pivot. Let's talk metabolism and the microbiome.
Ah, the gut connection. Always interesting.
This one's from July 10th. It's a preprint on Bioxov, that's pronounced bioarchive. From Motwani, as in colleagues at the National Institute of Immunology in India, RICTOR regulates an interspecies crosstalk that influences longevity through a novel methionine-Mitophagy axis.
Okay, breaking that down. RICTOR, that's part of the mTORC2 pathway?
Correct. And they found that losing RICTOR actually extends lifespan in C. elegans worms.
Interesting. How?
It seems to alter their vitamin B12-dependent methionine metabolism. This leads to higher levels of succinate, and that, in turn, activates mitophagy.
So they're proposing this novel pathway, methionine, B12, succinate, and mitophagy longevity, linking microbial metabolism through the B12 with mitochondrial health and lifespan.
Via a known nutrient-sensing pathway, mTORC2.
It's a neat mechanistic story in the worm,
Yeah.
but this immediately raises that big question, right?
Translatability.
Exactly. Worms to humans. It's always the hurdle.
Yeah.
The mechanistic detail is great, but applying it directly, especially the B12 effects, human metabolism, our microbiome, it's just so much more complex and context dependent.
So we need to see this outside of C. elegans before we get too excited.
Pretty much. Future experiments really should test, say, B12 restriction in mammals that are deficient in RICTOR.
And for the human link?
Measuring succinate levels in plasma for aged humans. That would be a really crucial step to see if this axis actually operates in us.
Makes sense. Okay, now here's one that should definitely grab the attention of anyone thinking about drug repurposing.
Always a hot topic in longevity.
This paper came out July 11th, preprint on MedHive, from Corley, MJ, and team at Weill Cornell Medicine.
Okay.
Semaglutide slows epigenetic aging in people with HIV-associated lipohypertrophy.
Semaglutide, the GLP-1 agonist. Ozempic, Wegovy, big name drug.
The very same. And in this randomized controlled trial, they found it significantly reduced epigenetic aging.
How significantly? And how measured?
Measured by GrimAge and PhenoAge clocks, pretty standard ones. And the reduction was substantial, between 1.4 and 4.9 years of age acceleration reversed.
Wow, in humans, that's quite something.
Yes, in this specific group, people with HIV-associated lipohypertrophy, they also saw reversals in inflammation, brain, and heart aging signatures.
So this is really the first solid clinical evidence that GLP-1 agonists can directly modulate epigenetic aging clocks.
Seems like it. Suggests real gerotherapeutic potential beyond just weight loss and diabetes, doesn't it?
It really does.
Yeah.
It forces us to think differently about drugs already out there. Maybe we should be screening more existing metabolic drugs for aging effects.
This is where it gets really interesting for biotech researchers, right? An existing, widely used drug hitting an aging biomarker in a human trial. That's a big deal.
Huge deal. But caveats are important here.
Always. What's the main one?
Well, the primary one is the specific cohort. People with HIV-associated lipohypertrophy. It's a specific condition. We don't know if these effects generalize.
Or if they last after treatment starts.
Exactly. Or if it's unique to this population due to their underlying condition or other treatments.
So next steps.
Definitely need long-term randomized controlled trials, but in non-HIV older adults. And we also need deeper mechanistic work. How exactly are GLP-1 agonists affecting DNA methylation? We need to understand that molecular link.
Right. Okay, another drug repurposing angle. This one's focused on vascular health.
Also critical for aging.
Published July 11th in Advanced Science, Haoni et al., from Hwasong University of Science and Technology. ATF3 deficiency exacerbates aging-induced atherosclerosis and clinical intervention strategy.
ATF3, activating transcription factor 3.
Yep. They found it's key for maintaining vascular health. It works through an ATF3 ATG7 autophagy loop.
Okay, an autophagy connection. Makes sense.
And here's the biotech angle. Terazosin, which is an existing alpha blocker drug.
Used for blood pressure, prostate issues.
Right. They showed terazosin stabilizes ATF3 mRNA. And this delayed vascular smooth muscle cell senescence, when cells stop dividing and cause problems, and it reduced atherosclerosis in aged mice.
So that's really interesting. A direct link from a druggable transcription factor involved in vascular aging to a potential clinical intervention with an already approved drug.
Suggests terazosin could be repurposed as a, what did you call it, a xenomorphic?
Yeah, something that modulates senescence. Not necessarily kills senescent cells like a senolytic, but changes their behavior. It's an attractive idea. However,
There's always a however.
the models.
The models. We have to be careful with mouse models of atherosclerosis. They're useful, but sometimes they overemphasize plaque formation compared to what happens in human vascular aging, which can be more about arterial stiffening and endothelial dysfunction.
So translation requires caution.
Definitely. For robust translation, you'd want to see terazosin tested in trials looking at things like arterial stiffness in elderly humans.
And maybe map out ATF3 better in humans.
Exactly. A single-cell atlas of ATF3 expression in aged human blood vessels would be incredibly valuable. See where it's really acting.
Good point. Okay, finally for this week's batch, let's look at a novel diagnostic approach using imaging. This could really change things for clinical trials.
The MRI aging clock paper, yes.
Yeah.
July 12th, preprint on MedRops, the Anti-Eye Consortium, and others from Columbia University. Multi-organ MRI digitizes biological aging clocks across proteomics, metabolomics, and genetics.
Quite a title. What do they do?
They developed MRI-based biological age clocks for 7 different organs.
Which ones?
Brain, heart, liver, adipose tissue, spleen, kidney, and pancreas. Quite comprehensive.
Impressive. And what do they find with these clocks?
What really stood out was that the MRI-based age of the brain and the immune system inferred partly from spleen, I believe, predicted mortality.
Okay, that's significant. Brain and immune again.
Yep. And the aging of these organs correlated with plasma proteomics data, allowing them to even prioritize 9 potentially druggable genes linked to organ aging.
So this offers potentially a non-invasive way to assess aging across multiple organs simultaneously.
Right. So what does this all mean for how we track aging in a more holistic, non-invasive way? This could be a game changer for clinical trials, couldn't it?
It could be. It's a powerful tool, certainly. And it reinforces the focus on the brain and immune system as key leverage points for longevity. But
Causality,
exactly. This data is associative. It shows correlations MRI organ age correlates with proteomics, correlates with mortality risk, but it doesn't prove cause and effect.
So its clinical utility isn't proven yet.
Not yet. As a predictive biomarker for, say, disease onset or for measuring response to an intervention, it needs much more rigorous validation. It's a fantastic research tool right now, but clinical prediction is another level.
So if it's associative, what studies do we need to actually prove its predictive power, make it actionable for biotech?
Well, you need longitudinal studies. Track people over time, measure their MRI-derived organ ages, and see if those ages predict who actually develops specific diseases later on.
And those druggable genes they mentioned.
For those, you'd want functional validation, things like CRISPR screening and relevant aging models, to see if manipulating those genes actually impacts the aging process causally.
Right, makes sense. Okay, so reflecting on this week, definitely some valuable work. Incremental steps, fascinating insights.
Absolutely. Reclassifying genes, new worm pathways, clinical hints for existing drugs, new diagnostic tools. It all adds up. Solid science.
But it feels different from last week, doesn't it? Last week seemed to deliver what many people were calling truly transformative work.
Agreed. The papers from the week before, particularly a couple of them, offered perhaps more compelling evidence, especially those tackling aging more directly in higher organisms or solving really fundamental biological puzzles.
So let's shift gears. Let's revisit last week, because one paper in particular really captured the imagination of the longevity community. It warranted, I think, an even deeper dive.
Yes, let's talk about that primate study. That was something else.
Okay, let's unpack this one. Because honestly, this is where it gets absolutely captivating for anyone in biotech aiming for real therapeutic impact. This paper, it genuinely feels like a potential model change.
Agreed. Set the stage.
Okay. June 27th, published in PubMed. Authors: Aisha Siddiq, Ismail M. Shakir Mowgli, from King Abdullah University of Science and Technology, COST.
Costs, China.
Attenuation of primate aging via systemic infusion of senescence-resistant mesenchymal progenitor cells.
And the core finding was pretty stunning.
Stunning is the right word. They engineered human embryonic stem cell-derived mesenchymal progenitor cells, SRCs, using FOXX03 to make them resistant to senescence.
Okay, engineered cell. Then what?
They administered these SRCs systemically, intravenously, into aged cynomolgus macaques, non-human primates.
And the results?
Significant slowing of aging across multiple organs. I mean, the list is incredible. Cognitive function, brain structure, bone integrity, immune function, reproductive system, skin, lung, muscle, hippocampus.
Systemic effects. Wow.
Yeah. And they backed this up with transcriptomic and DNA methylation aging clocks showing substantial reductions in biological age.
And the mechanism, did they get into that?
Partly, yes. They attributed some effects to exosomes derived from these SRCs, those little messenger vesicles. And critically, safety looked good. No tumorigenicity, no immunogenicity reported in the study.
So this is arguably the most compelling evidence we've seen for systemic rejuvenation in primates.
Right. The FOXO3 engineering seems key to bypassing senescence barriers, and the exosome part suggests maybe even a cell-free approach down the line.
And the multi-tissue impact.
Yeah.
That fits perfectly with the geroscience idea of targeting aging itself holistically. What's truly groundbreaking here is that leap from petri dishes or maybe mice straight to a non-human primate, showing effects across so many different systems.
It feels like science fiction becoming reality.
It does edge closer. As we connect this to the bigger picture, it really suggests we are moving towards therapies that could target the aging process itself, not just individual diseases. It challenges our models.
Absolutely. But it also raises that important question. Okay, it worked in these macaques. How do we even begin to think about this for humans? The practicalities, the scale.
That is the critical question for translation. Beyond the immediate science, what are the big hurdles for turning this into a human therapy? Scalability, manufacturing, regulation. It's complex.
So, given that huge potential, but also the complexity, what are the absolute must-do next steps for biotech wanting to pursue this?
Well, the challenges are significant, no doubt, but maybe not impossible. First, you need robust, scalable human cell manufacturing protocols. That's a major bioengineering challenge.
And safety long-term.
Absolutely critical. We need longer-term safety studies, probably in larger groups of primates, watching very closely for any hint of tumors or immune reactions over time.
Dose optimization too, I imagine.
Yes, finding that therapeutic window, maximum effect, minimum risk, and maybe exploring different delivery methods. Systemic 5 is one way, but maybe tissue-specific delivery could be better for some applications. Enhance efficacy, improve safety profile.
Fascinating. A huge undertaking, but potentially revolutionary. Okay, let's look at another major paper from that same period, end of June. This one focused on drug discovery. June 30th by Orksver, Shindipina and colleagues.
Ah, the Harvard Broad paper.
Yes. From Brigham and Women's Harvard Med Broad Institute. Molecular signatures of longevity identify compounds that extend mouse lifespan and health span.
This is a really neat approach.
Tell us about it. They developed this biomarker-driven platform.
Yeah, integrating gene expression data from multiple long-lived species, then using that to screen compounds in silico computationally before testing the hits in vivo in mice.
A pipeline for finding geroprotectors? And did it work?
It did. They identified two compounds, selumetinib, which is a MEK inhibitor, and vorinostat, an HDAC inhibitor.
Both known drugs used for other things.
Exactly. And they found these extended lifespan and/or health span in mice, both aged and young mice. Interestingly, there were some sex-specific effects, too.
That's always important.
Crucial. So the insight here is really about the approach, this systematic, high-throughput pipeline for finding geroprotectors. It's a big step beyond just testing things based on hunches. It changes the model for discovery.
Right. But the question it raises
Is translation, especially with those sex differences. How do you design human trials for compounds identified this way, accounting for potential differences between men and women? That aging dimorphism is real and often gets ignored.
a critical point. Okay, another breakthrough area. Telomeres. June 28th, PubMed.
Yeah.
Neha Nagpal and Sunit Agrawal from Boston Children's Hospital.
Telomere therapy. Always exciting, always challenging.
Title: Extension of Replicative Lifespan by Synthetic Engineered Telomerase RNA. They developed something called ETERC.
Engineered telomerase RNA component.
Okay. And the key thing is it seems to overcome some big hurdles with telomerase therapy. Delivery stability.
And the big one, the cancer risk, right?
Yep.
If you just turn telomerase on permanently, that's generally considered a bad idea.
Exactly. But here, they found that a single transient exposure to this ETERC extended telomere length and the replicative lifespan, how many times cells can divide in patient-derived iPSCs, and also primary blood stem cells.
Transient being the key word there.
Seems like it. That potentially mitigates the oncogenic risk because you're not leaving telomerase constantly active.
So this is really significant. A stable, effective way to get transient telomere extension.
Right.
That solves a long-standing problem. And showing it works in patient-derived stem cells, that gives it strong clinical relevance, especially for telomere disorders, maybe even more broadly for aging. A really novel approach.
Definitely feels like it. Okay, let's turn to dietary restriction mimetics.
Yeah.
Always a goal, right? To get the benefits without the fasting.
The holy grail for some.
June 29th, Bioarc Bihar Hernandez et al. From UCSF. Dietary restriction promotes neuronal resilience via ADIO.
ADL, what's that?
It's a steroid hormone. 5-androstenedione, 3017-diol. They showed it seems to mediate the neuroprotective effects and some health span benefits of fasting and CR.
How does it work?
Acts in the nervous system, reduces kynurenic acid, which is a neuroactive metabolite, and importantly, it seemed to suppress cellular stress caused by the APOE4 variant.
Alzheimer's risk gene. Okay, that's potentially very significant.
Right. But what's also notable is that ADIL didn't significantly impact lifespan in their studies. It was focused on health span, particularly neuronal health span.
Interesting. So if we connect that to the bigger picture, it prompts that question for biotech again, doesn't it?
Lifespan versus health span.
Exactly. Are we always chasing maximum lifespan?
Hmm?
Or is prioritizing functional resilience, especially neuronal resilience, a more achievable or even desirable goal for some interventions? ADIL looks like a potential targeted health span compound specifically for neuroprotection.
A more precise tool, perhaps?
Could be.
All right, our final paper from that June batch, a genetic mapping study, June 26th, Bioarc LS XEL Ella from EPFL in Switzerland.
Ah, the C. elegans QTL mapping study.
Exactly. Genetic mapping of lifespan and mitochondrial stress response in C. elegans. They used multi-omic data integration and quantitative trait locus mapping. That's like a genomic treasure hunt for genes linked to traits.
In this case, longevity and stress response.
Right. And they did this in a genetically diverse population of worms, not just a single strain.
Which is important for finding more broadly relevant genes.
And they identified a gene called FIP1 in worms, whose human homologue is FIP1L1 as a conserved modulator of the mitochondrial unfolded protein response, the UPRMT.
The cell system for dealing with stressed mitochondria.
Okay. And here's the really neat part. They found that FIP1L1 was also associated with metabolic homeostasis in the human UK biobank data.
Ah, connecting the worm gene function directly to human population data. That's powerful.
Really strengthens the case, doesn't it?
It does. The genetic mapping approach itself helps overcome limitations of using just one genetic background. And finding a conserved UPRMP modulator, like FIP1L1, especially with links to human metabolism, that makes it a really compelling target. It suggests the UPRMP is playing this key role in protein quality control.
That connection from the worm QTL all the way to the UK Biobank, that's always a strong indicator for potential translation, pointing to conserved pathways, really brings it full circle.
Agreed.
So, as we wrap up this deep dive,
Wow.
it's clear we've seen this fascinating contrast, right? This week, valuable, important, often incremental findings. Pseudogenes, worm pathways, clinical hints, diagnostics.
Solid building blocks pushing things forward steadily.
Definitely. But then you look back at last week, especially that primate rejuvenation study. That felt like a different category, a potential game changer.
It really demonstrated a different level of ambition, showing that systemic multi-organ rejuvenation in a higher organism might be within reach. That kind of work demands a model change in our thinking.
It really does.
And the sheer diversity of approaches we've discussed today is just incredible, isn't it? From meticulously digging into pseudogenes and microbial crosstalk
To developing advanced MRI clocks and pioneering these really complex systemic cellular therapies.
It's astounding. And for you, the biotech researcher listening, it just underscores the importance of being skeptical, truth-seeking,
rigorous.
absolute rigors, demanding that validation at every single step, no matter how exciting or groundbreaking that initial paper seems.
And that is exactly our mission here, keeping you, the learner, right at the forefront of these discoveries, helping you understand not just what's new, but why it matters, how it fits in, how it challenges or confirms our models. So as you go back to your work, you're thinking, here's a final provocative thought to chew on. What fundamental assumption in aging research do you think will be overturned next? Much like, say, the idea of a pseudogene being just junk was overturned. What's next? And maybe more importantly, how prepared are we as a field to embrace that next model change when it comes?